Hi everyone, I hope you're having a nice week. This week we're going to talk about our fourth brain tour, which are the coronal dissection cuts. So let's get started. So we went over this last week, but just to review again, we have gray matter and white matter structures, and the gray matter are pinkish, kind of gray nerve tissues in the brain and spinal cord, and these have all of our somas and dendrites in them, and these include regions involved in muscle control and sensory perception. Whereas white matter is white because of myelin, so all of the myelinated axons, and which causes faster conduction of signals and transports information over long distances. And now we have three cuts that are special to the coronal dissection. So we have the first cut that's anterior to the optic chiasm, the second cut posterior to the optic tract enters the brain, and the third cut which is anterior to the oculomotor nerves. So as you can see on the right over here, we have our first cut on the top, which is anterior to the optic chiasm. So to remind you, that's where our X is. And our second cut is posterior to the optic tract, so where the nerve from the PNS goes into the brain to the CNS, which is here in this middle cut, shown by dashes. And finally, we have the third cut, which is anterior to our oculomotor nerve right here. And here is the corpus callosum. This is the coronal cut view. You can also see it from the mid-sagittal dissection. We'll kind of go into that in just a second. But I wanted to go over what it looks like, because it looks different depending on what view you're looking at it from. So if we're looking at the corpus callosum from the mid-sagittal cut, it looks entirely different. I mean, this looks kind of like it's the top of like the midsection of like a deer's antlers, where like if these are our antlers and this is like that main section where the horns spring from or grow from, the mid-sagittal view is completely different. But to review, our corpus callosum is a white matter band that connects our two hemispheres together, so our left and right sides, which allows for communication between our left and right brains, or sides of our brains, and is our largest cerebral commissure. So commissures are white matter fibers that interconnect corresponding regions on each side of the brain, and surgically cutting the corpus callosum has been shown to control epilepsy. And epilepsy is an interesting disease that can have some problematic treatments, especially if you have such severe epilepsy that you have to have surgical treatments for it. I know that for really bad epilepsy, you can see occasionally, removal of the frontal lobe, and one of the earliest, most famous cases of this that some of you might have talked about in your other courses is the case of HM, removed most of his hippocampus and rhinal cortex and amygdala. I mean, so much so that anything that was left behind was considered a vestigial. And he was completely unable to form new memories from after the age of 15. Another treatment is lesioning the corpus callosum, and these all can have problematic effects down the line. Anyway, let's move on to our first white matter structure, the corona radiata. And this is just radiating white matter. So if we're referencing my metaphor from earlier in the coronal view for the corpus callosum, you see kind of the horns instead of the area right in the center where the corpus callosum is, kind of like the radiating horns. That is our corona radiata, as we can see with the red stars on the right. And this carries all, or nearly all, of the neural traffic to and from the cerebral cortex. And this is something you can only see from the coronal cut. You couldn't see this in horizontal or mid-sagittal or sagittal cuts. So moving on to the cingulate gyrus, which is a gray matter structure. It's located right above the corpus callosum. As you can see, I apologize for the blurry image. But our anterior cingulate gyrus is responsible for conflict control and processing and executive functioning, and the posterior cingulate gyrus modulates emotional responses as part of the limbic system with the amygdala. And thalamus, hypothalamus, and the hippocampus too, I believe. And this is a comparison of the cingulate gyrus from the mid-sagittal cut on the upper left, and on the bottom right we can see the cingulate gyrus on the coronal cut. So again, like I said with the corpus callosum, depending on how you're looking at them, they are entirely different looking. So this is something to keep in mind depending on how something is presented to you. These are the same structure but look totally different just from how we're looking at the brain. So this is our first cut, which is anterior to the optic chiasm, and this is what we would see with our first cut. So we have the lateral ventricle right here, and the caudate is in this area, and our putamen is here, or putamen. Really depends on how you want to pronounce it. There are multiple pronunciations for all of these things. And another thing to consider, again, our corpus callosum is right in the center. If you think corpus callosum, shares information between the two hemispheres, then it's always a safe bet to say something pinned toward the middle and it's a white matter structure, it's corpus callosum. Ventricle would be kind of like a hole because there's no liquid, because it's a preserved brain, and then caudate and putamen are our different, just kind of have to look for the boundaries between them. And looking at what we'll see in the second cut, which is posterior to the optic tract. So our first two cuts will pretty much have the same structures. The anterior cuts will have very small ventricles compared to the third cut, which you can see here with our blue pin. And this is just comparing our first cut to our second cut. So in our first cut, we have smaller ventricles, or only our lateral ventricles are visible, and the septum pellucidum is visible here, which is this little structure here. That was not my best circling, but it's really teeny tiny in this image. But that little flap right there, that is our septum pellucidum. But in the second cut, we have larger ventricles, and you can see the third ventricle, but you can't see the septum pellucidum in the second cut. And then just to review again, our four ventricles, so our interconnecting cavities that contain our CSF within the brain. We have the choroid plexus, which secretes our CSF, and which is continuous with the spinal cord and provides cushioning and buoyancy for the brain internally. And this helps to further protect our brains against sudden hits and jolts. So we again have the dura mater and the arachnoid mater and the pia mater to help us. But having the ventricles on the inside provides liquid cushioning and nourishment to the brains from the inside to the outside. And so moving on again, we're getting back to our coronal cut structures. We're going to talk about the septum pellucidum, which I was talking about just a moment ago. You likely won't be able to see this in mid-sagittal cuts, and it's because it's such a thin little flap of tissue. I kind of have it pointed out with this green arrow because it just separates the lateral ventricles from one another, and it's right below the corpus callosum, which honestly, it might be easiest to think corpus callosum right below septum pellucidum, just so you're not having to constantly look for this teeny tiny little flap of skin, which I know it can be kind of hard to see. But also know if you're looking at the first cut or the second cut, because you won't see the septum pellucidum in the second cut. And the only function that it serves is to separate our two lateral ventricles from each other. Here's another image on a different brain that more clearly shows, and I have it circled here. So we have our two lateral ventricles, and then we have the septum pellucidum separating it. So moving on to at least one of the structures that I find really interesting, the basal ganglia. And within the basal ganglia, we have the caudate nucleus, putamen, and the globus pallidus, which we will have a mixture of gray and white matter. The basal ganglia supports a lot of motor functioning. It's broadly implicated in Parkinson's disease because it's really strongly implicated with dopaminergic function, and also with skill learning. So the hippocampus is really involved with like episodic memory, but the basal ganglia is really strongly associated with skill learning, like learning how to draw something, or how to do the motor movements to sculpt something or make a certain dish. So another, connecting this to HM again, they would have him learn mirror drawing, so which is kind of complicated, where you can only draw an image by looking at your hands in a mirror to see what kind of memory he had left, since they noticed that he wasn't forming any new memory, so he couldn't learn any new skills. But since his basal ganglia remained intact, while his amygdala and hippocampus didn't, he was able to learn new skills, just not form new memories. So anyway, getting back to our basal ganglia, it's really responsible in voluntary motor responses, and damage to the basal ganglia will disrupt the ability to initiate voluntary movement, which is something that you see in Parkinson's disease. You see bradykinesia, akinesia, muscle rigidity, and resting tremors. And just as a reminder, so bradykinesia is the slowness of movement, akinesia is not moving at all. And if you've ever seen or had a family member that has Parkinson's disease, you will often see them develop these like very shaky hands or shaky arms, even when they're sitting down and resting. That is due to damage to the basal ganglia and other areas, and the striatum broadly. And it plays a deep role, as I said previously, in skill learning or procedural memory. And as just another note, you might see the phrase striatum or striatum in papers, and this is referring to specifically the caudate and the putamen, and it's striped in appearance, which is why it's called the striatum. We like to look at the striatum specifically because it plays a role in cognition, motivation, reinforcement learning, and potentially some types of reward. So it's a very interesting portion of the basal ganglia. And looking at the caudate nucleus specifically, this is one of our structures that make up the basal ganglia, and it's specifically responsible for movement, learning, and memory. And just note here that it is a pretty medial structure, meaning that it's closer to the midline. And then moving on to the globus pallidus, that is another name for pale globe. And it appears lighter than the caudate and putamen, and it's divided into our internal and external capsules. And it's involved with regulating movement, and it has an inhibitory or acts in an inhibitory manner that balances out excitation from other areas. So it broadly slows everything down and helps out to filter irrelevant stimuli from other parts of the brain. You have to know what the globus pallidus does, but for identification purposes, you won't have to find this structure. And damage to the globus pallidus, or having a pallidotomy, will cause damage to the globus pallidus and will cause involuntary muscle tremors. So moving on to the putamen, this is another part of our basal ganglia, which is a pin structure, and it's connected to the caudate. So remember, putamen plus caudate are often considered the striatum because of their striped appearance, and it's also involved with movement reinforcement learning, implicit learning, and a little bit of reward. And we have our internal capsule. So this is related to our aforementioned caudate nucleus. Excuse me, not caudate nucleus. I meant the globus pallidus. Excuse me, I'm so sorry. That's our internal capsule, which is a part of their globus pallidus, which is a white matter capsule surrounding subcortical structures, and it connects the striatum to the thalamus, which again, the thalamus is our sensory relay center to other parts of the brain. So to kind of help you identify our internal capsule, it's a band of white matter between two chunks of gray. So helps get information from one area that's motor related to another area that's motor related very quickly, because if it's a white matter structure, it's myelinated, so it's zooming. And now we're going to talk about our anterior coronal section. And we see here in our third cut image, then we can see our caudate and putamen, which again, if you can see the striped appearance here, this makes our striatum. So I think if you're going more medially, caudate, more laterally, then we have the putamen. We also can see the corpus callosum. It's our very, very strong midline structure here. And then we also have the lateral ventricles, which are separated by the, give it a second, septum pellucidum. So directly below our corpus callosum, or septum pellucidum. And this is another image, kind of to make it a little bit easier to see. And see the hippocampus, thalamus, our lateral ventricle, and our corpus callosum again. We're going to talk about the thalamus right about now. All right, so our thalamus, in the picture up top, that is our mid-sagittal cut dissection, and this is how the thalamus would look in the coronal at the bottom. So you can see they look entirely different. In the mid-sagittal cut, the thalamus looks more like an egg. And it's a little bit harder to see in the coronal cut. So I remember it by seeing like a more consistent tan color. But broadly, like I said earlier, our thalamus is our sensory relay system to get sensory information from the different parts, like our hands, our eyes, nose, everything else, and sends that information to various parts of the cerebral cortex and midbrain. It's also considered the gateway to consciousness, and damage to the thalamus can lead to a permanent coma because it partially regulates sleep and wakefulness. So if we damage the thalamus, then we're potentially setting our sleep regulator to always sleep, no wake. And there are more than 100 different nuclei within the thalamus, and there are different functions within each nucleus. Pathology to the thalamus varies depending on the site and extent. It's a very interesting structure. I mean, we think, oh, it's just a sensory relay. It does a whole lot more. So our next gray matter structure is the pineal body. And there is only one pineal body, and I said to remember Descartes because he is claimed or has been cited as saying that he believed that the pineal body or the pineal gland was the seat of the soul in the brain. And it's responsible for the secretion of melatonin, which helps to induce our sleep-wake cycles and controls our circadian rhythm. And it's posterior to the to the thalamus. So you can see in the mid-sagittal cut, it's this little bump right behind the thalamus, and the coronal cut, it's still posterior, but it's a little bit different compared to looking at the mid-sagittal view. So moving on to one of our more famous structures, the hippocampus. It contains both gray and white matter and is often kind of described as looking like a seahorse. It's located in the medial temporal lobe and has lots of functions in learning and memory. This is one of the main structures that we learned about when you talk about HM. Not to bring him up again, but definitely quite important case. And there are plenty of others as well. Clive Wearing was another case of hippocampal damage. I have a video about that. Actually, I will put it in the description of the video, and I'll leave it on the as a Blackboard link as well if you would like to check it out. And I highly recommend that you do. It's really interesting to see how Wearing functions after his accident, after he sustained hippocampal damage. He had a viral encephalitis, I think, like brain encephalitis, when he did not catch it fast enough, so he had major damage to his hippocampus. Regardless, the hippocampus is involved in learning and memory, so specifically episodic memory, so times, places, context of who, what, where, when, why, detecting novel places, stimuli, and events. Also very special for learning spatial memory, which is what we use the Morris Water maze for, so learning how to navigate a maze. So over time, your episodic memory should get more and more robust, and it's assumed that in healthy mice, it would take them less and less time to complete the maze if they don't have hippocampal damage. And lesions result in severe anterograde amnesia, which means that you can't create new memories after the damage or whatever caused your amnesia, whether that's sickness or some sort of brain damage. So long-term memories before the event remain intact, but you can't form any new memories after. Again, a perfect example of HM, since he couldn't make any memories after 15 years old. And our next view here, you can see the hippocampus and the thalamus and our coronal cut. And finally, we can see the lateral ventricles with our blue and green pins here through the coronal section. They definitely look quite different, but if you can imagine them filled with CSF, then they would be quite large, relatively speaking. And we have our third ventricle in the coronal view. Again, shown by our green pins here. I think that they kind of surround our midbrain nuclei quite well. And to round it off, just to review again, our coronal cut, so we have our first cut is just anterior to our optic chiasm. The second cut is posterior to the optic tract. And our third cut finally is anterior to the oculomotor nerve, And here is a little review of each of the structures that you can see in each cut. I'm not going to read all of them, but this is a wonderful reference. And you might not be able to find each structure in each cut, but generally speaking, you should be able to see these in each coronal cut. And here is another image comparing our first and third cuts. You can see hippocampus, our thalamus, the lateral ventricles, or again, our corpus callosum, but you should see the corpus callosum in all of the coronal views. And again, make sure to take a look at this and maybe do some comparisons with the mid-sagittal lecture from last week. But that is it for this week. Thank you everyone for tuning in. And our attendance word for this week is going to be coronal. So from the title slide, coronal. Please let me know if you have any questions. But please take care of yourselves, and I hope you have a wonderful week. Have a good one. Bye.